There we go. Okay. So we're joined by Dr. Merantha Moodley and Trevor Khaba, who they are both from the Ndhlovu lab at AHRI in Durban. We're seeing, yes, now we're seeing the correct part of your presentation. Merantha is a postdoc and Trevor is a senior technician in the lab, and they're going to speak to us about how they use microscopy in their interrogations of HIV immunology. Over to you two.
Good afternoon, everyone. Thank you so much for this opportunity. My name is Merantha, and this is my colleague Trevor. We are from the Ndhlovu lab at the Africa Health Research Institute, and we are really excited to present the trends and advancements in microscopy, particularly in HIV and immunology. In terms of a global overview, there is significant amount of progress that has been made, particularly within the African continent, which is known to carry the greatest burden of HIV. This progress is seen both in a decrease in the number of new HIV infections as well as the number of AIDS-related deaths since 2010 until 2022. However, a significant challenge still persists in terms of not all people living with HIV are on antiretroviral therapy, and not all people living with HIV are virally suppressed. This challenge is also seen on a global scale, in which there are only 71% of people living with HIV who are successfully virally suppressed. Now, why is this still a problem? When people living with HIV are on suppressive antiretroviral therapy, they have viral suppression, and they usually have undetectable viral loads. So why then are we still searching for an HIV cure? This is because HIV DNA can integrate itself within DNA of our very own immune cells. This is not that harmful when it is latent and inactive. However, when patients have antiretroviral therapy interruption, viral rebound can occur. The mechanism behind this is that the previously inactive DNA of HIV can become activated and produce viral RNA and viral proteins. The source of these reservoir cells is a multidimensional landscape. This landscape has been reviewed by the International AIDS Society's Global Scientific Strategy in 2021 as research priorities for an HIV cure. One of the key goals to be addressed in the next 5 years includes understanding HIV reservoirs. Within the HIV Reservoir Working Group of the International AIDS Society is our mentor, Professor Zaza Ndhlovu. Professor Zaza is born in Zambia within Africa. He obtained his PhD in molecular microbiology and immunology at the Johns Hopkins University and went on to pursue his postdoctoral research at Harvard. He became a faculty member at the Ragon Institute at Harvard. And what is so astonishing and remarkable is that he chose to come back to Africa, where he relocated his research program to South Africa, the epicenter of HIV, at the Africa Health Research Institute. And he also maintains strong affiliations with other prestigious institutes as well. Here, our group investigates HIV reservoirs in tissues such as the lymph node, where we have done extensive research, and we've recently began investigating the gut tissue as well as post-mortem human brain tissues. Within our research team, we have a diverse group of young and ambitious scientists. And as part of the group that undergoes microscopy analysis, we form part of the Ndhlovu Lab HIV Cure Group, and our broad aim is to spatially detect and quantify the replication-competent HIV within tissues, and phenotype and identify the genetic composition of the key immune cells in which the virus persists within each anatomical compartment. This is with the ultimate goal of discovering novel insights that can inform HIV cure strategies. One thing that Professor Zaza is incredibly passionate about is contributing towards scientific capacity building that is needed to meet the current and future African medical challenges. I now hand you over to my colleague Trevor, who will tell you more about this.
So in 2015, our lab initiated the collection of lymph node tissues with the objective of exploring the HIV reservoir and HIV-specific immune responses within human lymph node tissues. We established a functional collaboration with the surgical team at the Prince Mshini Hospital in Umlazi, facilitating the acquisition of tissue biopsies from individuals living with HIV, as well as uninfected participants enrolled in the study. Building upon the success of this collaboration, we expanded our inquiries to encompass additional tissue sites. In 2002, we began the collection of gut biopsies to investigate the HIV reservoir and HIV-specific immune responses within human gut-associated lymphoid tissues. To facilitate this increase in tissue collection, we enlisted the service of the Durdock Hospital in Durban for the collection of excisional lymph nodes and gut biopsies from recruited participants. These tissues are received by us in the lab where they are fixed and embedded in wax. They are then sectioned to create FFPE tissue section slides. So additionally, with the expansion of our research capacity in histology, we initiated a collaboration with the Stain Lab at AHRI, through which we received post-mortem brain tissue sections. So our lab has access to a diverse array of imaging technologies. Currently, the foremost amongst these has been our wide-field fluorescent microscope system, which has served as the primary tool in our imaging work. Additionally, we are in the unique position of being the only facility on the continent to house the GeoMx digital spatial profiler, a cutting-edge technology that enables high-resolution spatial and molecular analysis of tissue samples. Also, as part of our affiliation with the African Health Research Institute, we also benefit from having access to several of the microscopy core imaging instruments, such as the Hamamatsu Nano Zoomer scanner and the Nikon Eclipse inverted confocal microscope. So the GeoMx DSP from NanoString offers the capacity for very high-plex spatial molecular analysis of RNA and protein within tissue sections. It provides spatial information alongside molecular analysis, allowing researchers to understand gene expression patterns within the natural biological context of cells. It overcomes the limitation of traditional single-cell RNA-seq by providing a spatial dimension to gene expression data. This technology offers a powerful solution for spatially resolving gene expression profiles within tissue samples, facilitating a deeper understanding of biological processes, cell phenotypes, and disease mechanisms. So with support from the Bill and Melinda Gates Foundation, our lab has also established the Center of Excellence for Spatial Omics Research in Africa, also known as CISORA. CISORA offers a variety of educational and training opportunities to aspiring researchers, emphasizing hands-on learning and project-based approaches. These initiatives aim to equip participants with the knowledge and skills for utilizing, advancing, and applying spatial omics technologies in the investigation of diseases within their native tissue contexts. Recently, CISORA welcomed its first intern from Harvard University, who engaged in a 9-week immersive experience focused on utilizing CISORA's GeoMx technology to elucidate the transcriptional profile of HIV-infected macrophages within lymph node tissues. Furthermore, CISORA organized its very first annual in-person workshop last year, comprising of a theoretical session and practical exercises covering the entire spectrum of spatial omics research workflow. For additional information on CISORA, I encourage you to visit the official webpage shown on the screen. So in 2018, our lab invested in an Axio Observer imaging system Several factors influenced our decision to choose this particular system. The axial observer enabled us to capture entire tissue sections with significantly enhanced resolution and speed compared to previous scanners that we had access to. A need to expand our research question prompted us to upgrade to the axial imager, which is equipped with an additional 8 custom filter sets. And these additional filter sets expanded our capacity to extract more information from each tissue section. And it will empower us going forward to tackle a wider range of research questions. So as previously stated, our tissue collection currently encompasses samples from 3 anatomical sites: the lymph node, the gut, and the brain. While most of our published research has focused on lymph node tissue, we also have published data on the gut, and our investigations into the post-mortem brain tissues has yielded promising results. And so I will speak briefly on some of the lymph node work we've done, and Merantha will speak more on the new research we are doing involving post-mortem brain tissue. So one of the core areas of interest in our group has been looking at HIV reservoirs. In line with other research groups, we have published data suggesting that despite the initiation of early antiretroviral therapy during HIV infection, the virus continues to persist in germinal centers within lymph node follicles. Moreover, there is a continued ongoing viral replication in these sites despite successful ART therapy, which poses an obvious challenge in achieving complete viral elimination. So a possible contributing factor to the persistence of HIV in the germinal center is the spatial segregation of CD8 T cells that are the primary adaptive cell tasked with eliminating infected cells in the immune system. Understanding why the spatial segregation exists might help in understanding these sites and how to disrupt them. Moreover, we observe that there is some level of recruitment of CD8 T cells to these sites in response to viral protein. However, these follicular CD8 T cells have a weakened cytolytic ability. And from data we've generated from our spatial omics multidimensional analyses, we believe that they might have an altered phenotype that is geared more towards regulatory and anti-inflammatory activities that further promote the persistence of virus in these sites. And so we are trying multiple approaches in our research, and they are leading us down interesting paths of inquiry and hopefully towards meaningful answers.
Okay, so just a little summary on the brain research that we are doing. I joined the Ndhlovu lab last year in April, where I began my postdoctoral journey. And these images were taken by our immunofluorescence microscope, showing the magnitude of the resolution that we are getting, even without oil immersion microscopy. These were acquired on the 20x objective. So this shows HIV gap pol RNA in green, which is an incredibly minute signal. And we are able to see the HIV RNA in the merged image co-localizing with IBA1 positive microglia in the brain. Microscopy is also useful in identifying cells which express both HIV RNA in green as well as HIV protein P24 in white. These are within GFAP positive astrocytes in the cerebellum. So even in a tangled mesh of complicated networks of brain cells, we are still able to detect signals as minute as HIV RNA, and I think that's incredibly wonderful. Using the integration of going back to the basics, such as H&E staining, has been incredibly useful when investigating brain tissue in the context of HIV reservoirs. Unlike lymph nodes, which have germinal centers that are easily used to detect HIV protein and RNA within these regions, I've noticed that within the brain, HIV RNA and protein stains differently within these regions. So I used H&E imaging to help me stratify the subregions within each region of the brain. and I layer this onto my immunofluorescence staining so that I could demarcate the different regions and understand the spatial localizations in which HIV RNA and protein persist. So essentially, we are using different techniques in conjunction with one another to enhance our understanding of what we are seeing through the microscope. In terms of quantification, we generally use the StrataQuest image analysis software from TissueGnostics. This software involves processes of nuclear masking, which masks all nuclei within the tissue, as well as cellular masks, which are able to mask even cells with complex morphologies. Whether these cells have spaces around them or whether they are in clusters, we are able to successfully and accurately mask these cells. We also recently purchased the Halo imaging software from Indica Labs to be able to quantify the minute RNA and DNA signals within the brain. I would like to thank you to the IDM, to Zeiss, as well as ABIC for this opportunity, and I'll hand you over to Trevor to do the concluding acknowledgements.
Thank you. I would like to just acknowledge our program and lab PI and director of SOSORA, Professor Ndhlovu, for his leadership and mentorship. Our Ndhlovu lab team, including our collaborators, who work tirelessly to ensure that we are continuously generating and publishing data to contribute to the collective knowledge pool. The study participants and staff involved in providing us with the invaluable tissue samples that are at the core of the work that we do. The STAIN Lab for their contribution to the postmortem study, the Bill and Melinda Gates Foundation for their funding for SOSORA, the Reagan Institute for the funding for the purchase of our imaging system. And I would also like to acknowledge all of the institutions that have contributed to making all of this possible: the NIH, HHMI, HPP, AHRI, Sante, and UKZN. Thank you.
Thank you very much. That was great to see the range of technology that you're using there to pursue your questions. I'm going to open it up to the in-person audience here if there are any questions regarding what you've heard. Otherwise, any questions online? Okay, so I'm going to, we've heard that our speaker from Ghana should be joining in a few minutes. So I'm going to, oh, we have an in-person question here.
Thanks to the two of you at the AHRI. A question for both of you joining Zaza and Ndhlovu's lab. What has it been like getting into the microscopy for the first time, getting to grips with the techniques and the methodologies and approaching optimization and things like that? Can you give some insight into that, please?
I think for me, it was very exciting. My background was in like cloning and expression molecular work before I started here, and you know, I've learned a lot. I mean, now we are, you know, not only just doing spatial imaging, we're now linking that to genomic work, and so I really find everything that we're doing here very exciting.
And I think with me, it's pretty much similar but different experience. So I did come from a microscopy background where I did bright field microscopy, mainly just the immunohistochemistry dab staining. That's the only type of microscopy I did before joining the lab. So I did have some experience in terms of tissue sectioning and the staining process, but the amounts of resources we have and the value of these resources and the novelty of these resources is really incredible to have the opportunity to work with these resources that we have. And I've been trained by Trevor, and here I am, not even a full year later after my postdoc, I'm co-presenting with him. which shows, I think it makes me feel proud to be a part of the team because it shows that no matter what level per se you are, we all really work together. And at the end of the day, we're all sort of on the same plane, always learning from each other, teaching each other what we discover that could help.
That's useful and a good response. And I'd like to follow up on that. And this, I think, is a question I can open up to our in-person speakers as well. But if you could take a moment to talk through how you've chosen the technology that you're using, especially in a setting where you have access to so many different technologies. Ryan touched on it a little bit in his talk, but I guess to start with Trevor, Merantha, given the range of technology that you have access to, what sort of decision tree that do you use to choose which system you're going to use for any given experiment?
Okay, so in terms of my project, when I joined the lab, previously the lymph node tissues were very widely investigated, and most of the team had a lot of experience in the lymph node. But the brain research was still relatively new, and we were trying to understand how to best use microscopy to fully answer our research questions. In terms of H&E staining, I feel like that's something I personally integrated much more into the brain reservoir project, because when it comes to the lymph node, for example, immunofluorescence staining can be used on its own because there's a particular marker for the germinal centers that can be used with multicolor immunofluorescence to detect the zones in which HIV persists. Now, unlike that, when it comes to tissues like the brain, where there's no particular zone per se, and you still have to classify the regions, that's where H&E, I chose to incorporate H&E as my fundamental aspect to help me understand what are the different regions of each brain site, because these regions can house different types of brain cells and the cell bodies of neurons versus areas that do not have the cell bodies of neurons. So in terms of the GeoMax technology, which we have access to, that is a future plan of my project. But in order to get to that stage, I would first need to use the immunofluorescence to detect the most prone areas to HIV persistence, because we can't be looking blindly in the GeoMax without elucidating that closely before we go and assess the genetic information.
Great. Thank you so much. Anything to add to that from our in-person speakers? In terms of choosing how you've chosen the technology that you've chosen with regards to imaging?
I mean, I think, as I mentioned earlier, in our context where resolution is not the primary goal, as imaging, ease of imaging and robusticity for sample is what drives our motivation. I think the simpler technology in wide field fluorescence is fairly robust to those things. I guess it just depends on what your application is, what your requirements are in terms of tissue depth and resolution required is, yeah. But I think for our purposes, even in microbiology more generally, a lot of people would lean towards confocal because of the increased Z resolution. But in our experience, I think with the bacteria being so thin, we can get away with a lot just using a wide field fluorescence, actually get really high quality images with good resolution.
Great, thank you, Ryan. I'm aware that we have a question online, and then our third speaker has joined us for the day. So I think this is for you, Trevor and Merantha. Do you think the HIV viral reservoirs in some tissues is related to how well the ARVs penetrate into the tissue? For example, the blood-brain barrier hindering penetration of ARVs. And is this something that you will be researching?
Yes, definitely. In fact, the blood-brain barrier was something that we just discussed and was advised by Professor Zaza for us to start looking into. Yeah, so definitely there is a lot of heterogeneity and diversity across the tissue landscape of HIV reservoirs. That's been a challenge to HIV cure. And as we gain more understanding into each tissue and its own specific mechanism of antiretroviral therapy penetrance and viral reservoir persistence, can we get steps closer towards eradicating these reservoirs towards an HIV cure?
Great, thank you so much. I'm aware that we're running a little bit behind time, but I'm thrilled that our third speaker, Dr. Yao Aniwe from WACBIP in Ghana has joined us. So I'm going to, Yao, I'm going to invite you to share your screen with us.